going to flip my camera around so this makes a little more sense. So in the foreground here, I've got some fruit flies from the genetics lab, but in this space, during the summer, we make use of that to collect specimens, make vouchers, and I've got a microscope to do morphological analyses. So I've been involved on a lot of questions related to invasive species. So Japanese knotweed is a recent project. We've also worked on Phragmites, hydrilla, which is not a big one in Wisconsin just yet, but get samples from other parts of the country, and we've studied that one. And I sort of have a general functionality to be able to do genetic identification, and I've kind of branded myself as somebody who can use DNA to augment regular identification methods and give people a positive identification on unknown plants that might be difficult to identify with morphology, or new ones that are unfamiliar, or for whatever reason just kind of give somebody a little more confidence in their plant identification. And that's also something that I've involved students on. So I get students who are interested in getting some lab research, and they kind of come to me looking to get experience doing DNA work, PCR. We do DNA sequencing here, gel electrophoresis, and they can do this in a context that's productive and meaningful. So we answer questions of interest to the scientific community, to the land management community, and so on. So I have training in aquatic plant systematics. That's kind of my background. And then basically with a DNA add-on to that. So aquatic plants are my specialty, but I'm pretty much open to whatever questions come my way. And those that tend to be, aquatic plants, invasive species, a little bit about ecology, and so on. Have that space behind me is the research lab where we do the DNA work. I've also got this space, which is a prep room adjacent to my lab, where we can do plant growth experiments. So we have growing trays, media, and so on. And then I have kind of a modest, just a kind of a standing collection of plants that I can work with. Just involve students on other aspects of plant growing. In this space here, I've got a student who's currently doing a growth experiment on Azolla, the water fern, doing different nutrient media that they can grow in. So lots of space here. I run through 5 or 10 students a year doing research, and lots of opportunities for them to work in the lab. Of course, when the weather gets nice, and sometimes during the summer, we get involved working outdoors, and involve students on plant habitat surveys, so identifying plants and making inventory lists of properties. We worked with the Nature Conservancy on some projects where they have newly acquired land and they want to know what plants are located on there, terrestrial and aquatic plants. So I've got background in plants. I don't always have students who are interested in plants, and I'm pretty happy to work with them. Whatever they come in interested in, we can work on that. Microscopy, we do light microscopy here, just sort of a dissecting microscope setup. We also have the ability to do electron microscopy. I don't know if I'll be able to show that to you, but I've got some samples in the cabinet here. These are gold-coated samples of seeds that were collected from wetland soil cores. And as part of documenting those, we have run them through the electron microscope downstairs. So lots of chance for students to get involved.